So I have the privilege of introducing Dr. Alan Beggs. I'll read you a bit about his bio. It's just relatively impressive. But I want to say, first off, that Alan has been a great advisor, personal advisor to the foundation, a personal advisor to me as we started off on the journey of trying to figure out neuromyopathy and sort of the least path of resistance as we sort of taken this, what was a little tiny ship, and now built this bigger, massive thing, and try and produce, some results. Alan is one of the foremost researchers in Nemelin, and in fact, in many congenital myopathies, certainly in the US, if not the world. But I will say also, he's also a person that has dedicated his whole life to supporting people with myopathies, and in addition to his work, I will say that he's been accessible to us. He responds to texts, he takes my phone calls, and he's been helpful. He's actually helped our foundation and told us when not to spend money on something that might be wasteful. So, how often do you hear a researcher say, don't pay for that, right? So, I think that in that way, he's been supportive of the foundation, he's been supportive of us. And I think he's made AFBS a better organization. He's definitely helping the world of NM. To read you a short bit about some of his accomplishments, he's the director of the Manton Center of Orphan Disease Research at Boston Children's Hospital, and Sir Edwin and Lady Manton Professor of Pediatrics at Harvard Medical School. Following his studies at Cornell, he got his PhD in human genetics at Johns Hopkins. I'll skip through some stuff because he just has done so much. Throughout his career, he used the tool set of human molecular genetics to study normal biology and disease processes in a variety of diseases, with a particular focus on NM. Over the years, the Beggs Lab for Congenital Myopathy Research has contributed to the discovery of 7 of the 10 known genes for NM. Beggs and his team have been active in both clinical and laboratory studies to define the progress of NM and develop potential treatments. He's been a standing advisor of our board, probably for 6+ years, if not more. He also, I think his lab has fathered more folks in the research of NM. Mike Lawler was here, Kathy North, and the list probably goes on and on. Thank you.
Thank you, Mark. I thought you'd have a shot. It's great to see everybody here. It's nice to see everybody back who I've met in the past, and I'd love to see some new faces out there. So I'm going to try to give you an overview. This is, I guess, the first scientific talk of the conference, and just kind of define what are congenital myopathies, what is Nemelin myopathy, tell you a little bit about what we know about it. If I say anything to go too fast that you miss, please don't hesitate to raise your hand and ask for clarification or questions. And for sure, I'm going to leave time at the end to talk about, have open questions and so on. Also, I'll be here throughout the conference. I'll be downstairs with the natural history study for a lot of it, but don't hesitate to come up to me and grab me by the elbow. And just ask me a question anytime throughout the conference. I love, explaining what we do to people and trying to help you understand what it all means. So this morning I'm going to cover at least hopefully the first 3 of these 4 points. Just talk about, explain what are congenital myopathies. You've probably heard this term. Some of you have heard muscular dystrophy. You recognize that Nemelin myopathy is not muscular dystrophy. Let's explain specifically what makes Nemelin myopathy as opposed to myotubular myopathy or congenital fiber type disproportion, or some of these other phrases you may hear bandied about. Then I'll tell you a little bit about the genes. As Mark mentioned, we now know of 10 genes primarily that can cause Nemelin myopathy. Everybody doesn't have mutation in all 10, but they would have alteration in one of these, we believe. And there are probably still a few more out there we haven't discovered yet. And then I'll end up this morning by talking about the biology. Knowing what the genetic defect is that causes weakness doesn't completely tell us why muscles are weak. And that's very important to understand because, as you'll see throughout the conference, learning about the mechanism of weakness can actually be really critical and important for understanding potential therapies, ways to treat the weakness, and that's really ultimately what we want to do here. And then I doubt I'll get to it today. I have a few slides at the end about zebrafish, how we're using them. If I don't do that now, though, we'll talk about those tomorrow, and I'm also going to talk about gene therapy and CRISPR, which I understand a number of people have been asking about. So, wait till this
It just takes a
turns
second.
on. Yeah. Is this on? Okay, good. So I'm going to start by quickly going over the basics of muscle contraction and structure, just to kind of orient you. And this is from an animation that's actually on our website. The URL will show it later if anybody wants to know, or if you Google search for my laboratory's website, you can find this. But just to remind everybody, here's a baby shaking his rattle. There's a muscle here. Mark can tell us what muscle this is.
Biceps.
The biceps. And Mark knows better than me. The muscles are broken down into muscle bundles and then individual fibers. And then from that, you've got a single muscle cell coming out here. And the muscle cell contains a series of repeating units that we call the sarcomere. And blown up, the sarcomere extends from one of these vertical gray bars to the next. These are all given names, so these vertical gray bars are called the Z lines or the Z bands. You can see that here. And then what actually allows muscle to contract is the fact that this whole sarcomere shortens because something called a thick filament, shown here in red, which contains a protein called myosin, some of you may have heard of, has these heads, these little moving balls that grab onto and pull the thin filament, and that's what this gray stripe is here. So this is really looking at the machinery of muscle contraction at the molecular level. So each of these little balls and heads and so on, that's a single molecule. So you can't even see that in the microscope, but we can kind of see the whole thing put together.
That's it.
So this thin filament is not just one thing. It's actually a complex structure, and that's shown here and blown up in detail. And one of the major components that the myosin grabs onto that you've probably heard of is actin, and that's these gray balls. So each of those gray balls is a single molecule, so single protein. So when we talk about genes and proteins, each gene has the information to make one of these gray balls, one protein copy of actin, and then they all kind of align together in a filament to form this long structure. But it's not just actin that's present in the thin filament. There are other proteins that help regulate it. So one of them, many of you have also heard of, is called nebulin, and that's this big green stripe. So nebulin, just to go back for a second, begins here at the Z line, and it extends out to the end of the thin filament. And you notice that all the thin filaments in this sarcomere are the same length. So nebulin is one of the proteins that helps control that. You want the overlap for force to be generated to be even and maximal between the red myosin and the gray actin so that the myosin can grab onto the actin and pull. And so in order to, for this to work efficiently, nebulin, which spans the length of this, begins at one end, ends at the end here, and helps determine the length. Now, obviously muscles are voluntary, so when we initiate a muscle contraction, it's because our brain decides it wants to do it. Electrical impulse travels down our spinal cord to our nerves. The nerves are the wiring system of our body. They go out to the muscle. And what happens at the end of all of that is when that nerve fires and tells the muscle to contract, calcium comes rushing into the muscle cell. And that's the signal that tells the muscle to contract, or the sarcomere to shorten. And the way the calcium works is it binds to this protein called troponin. And then troponin in turn is bound to this protein tropomyosin. So don't worry too much about the details here, but I'm throwing out all these names because some of you in this room have alterations in some of these various genes. So some of you, as you'll see, will have an alteration in the gene for tropomyosin, some in nebulin, some in actin, and some in some of these other genes. So what happens when the calcium binds to troponin, it changes its shape and moves. That makes the tropomyosin move, and it actually slides out of the way and exposes the actin so that the red myosin over here can grab onto it. And that's what causes this contraction that I was showing you in the previous slide. All right, so that's just to give you a little bit of background for what these various proteins are. And let me come back now, and I'll tell you a little bit about congenital myopathies. But yes, I'll take a question, please.
Can you clarify some more about the mechanics of the pooling? Because it's not clear from the animation how the physical pools.
Right, so the question is, can I clarify a little bit more about the mechanics of pulling and how does that actually work? I was actually in a meeting in Madison, Wisconsin about a week or two ago with about 300 people just focused on exactly this question. They were thinking about the cardiac muscle and the skeletal muscle. How do you mean clarify the mechanics? I mean, what happens is that the myosin protein energy comes from a molecule called ATP. Many of you have heard about that. ATP is made in mitochondria, which are the powerhouses of the cell. And the myosin is like a spring. It's loaded, and it's actually cocked, waiting to pull. And the only thing that prevents it from pulling is tropomyosin being in the way. And when the calcium comes in and everything moves, tropomyosin moves out of the way, it exposes the actin. Now the myosin can grab onto it. And because of the way the energy of the molecule is, with the ATP loaded up, it wants to grab and shift its shape. And then as soon as it's done that, it releases and everything goes back. And so it's kind of like that each one moves a little bit. And what's happening in milliseconds, nanoseconds, is that this is going over and over again, kind of creeping a little bit like the legs of a centipede. Is that a sort of answer? Yeah. There's a lot more biophysics that goes into it if you ever want to find out. But frankly, a lot of that's even beyond me.
Can I try for a second?
Yeah.
So,
So the way I think about it is when a muscle contracts, whether it's a breath or whether it's a simple movement of your arm, your elbow bending, the bicep muscle has to shorten, right? And the way he was describing is that your brain tells the muscle to shorten. And so what happens is the calcium that flows to the muscle basically lets this, for lack of a better term, a magnetic process happen, right? So it exposes the magnet, if you will, so that now all of a sudden the myosin can bind to the actin, and then, like you said, the shape changes, and it makes it turn over and then release, and then grab a new one and turn over and release. And the problem that we're seeing in those with NM, correct me if I'm wrong, Alan, is that the actin or the nebulin may not be at the proper length. And so you may not get the overlap of actin over myosin. So if, hold on. So if I have actin and myosin, if they can't even reach each other, how can they make a contraction happen? So for those that are severely affected, you don't even get connection happening. And for those that are more mildly affected, you're getting some connection, so some reach, but you're not getting a lot of overlap, so you're not getting a lot of those magnets connecting.
Right, okay, thank you. And actually, I'll get into 3 different mechanisms where this may happen towards the end of the talk. A little bit differences, whether it's something like tropomyosin or nebulin or actin. So congenital myopathies, hypotonia, many of you know, means floppiness, low tone in the muscles. So a floppy baby kind of like slips through a parent's arms when they're lifted up. And congenital, as the name implies, is at birth. Bio means muscle, pathy or pathology is disease. So these are simply muscle diseases present at birth. And they present, when we say primary hypotonia and weakness, that means that the cause of that weakness is intrinsic to the muscle itself. It's a primary defect. If the defect is in your brain or your nerves, then that will be a secondary weakness in the skeletal muscles. These conditions, unlike muscular dystrophy, are considered to be relatively non-progressive or less progressive. That doesn't mean people don't change over their lifespan, but in contrast, say for muscular dystrophy, a boy with Duchenne dystrophy may walk fairly normally when he's 3 or 4, may start to stumble and have difficulty walking in when he's 5 or 6. Typically needs a wheelchair by the age of 11, and a few years later often needs additional respiratory support. So that's a highly progressive condition, and all the boys with Duchenne dystrophy follow that. In myopathy, there's a lot of variation, as you know, and some people walk for a good part of their lives, some people never walk, some people will walk and then later in life they'll need increasing help getting around. So it's good. When we say relatively non-progressive, people will say, oh, well, I either am progressive or I'm not. There's a lot of variation, but it's distinct from the muscular dystrophies. As you know, there's wide variation, and you can see that here among all your friends. It's genetic. It's considered to be relatively rare, but in fact, it's the most common of the congenital myopathies. Congenital myopathies actually account for maybe 1/10 of all of the patients that are seen in the Muscular Dystrophy Association clinic. So muscular dystrophies or spinal muscular atrophy are somewhat more common causes of muscle weakness. The changes that define these originate within the muscle fiber, and again, that's what makes them a primary myopathy. And there are distinctive and specific changes that define these. So the different congenital myopathies are given names, and Mendelian myopathy is one of them. But just to quickly illustrate what we mean by some of these terms, these are 4 different staining patterns. These are all 4 muscles from an unaffected individual stained with different dyes in order to look at different aspects of the muscle. And I'm just going to take you through a few examples of how each of these can be unusual and define a different congenital myopathy. So here, on what's called an H&E stain, you see a muscle fiber with a nucleus around the outside. It's a small blue dot is the nuclei. And here is the biopsy from an infant who has something called myotubular myopathy. And that's because the first thing is the muscle fibers are much smaller relative to the size of the nuclei. The second unusual feature is that the nuclei are found in the center of these muscle fibers, and they look like myotubes, which is a normal stage of muscle development during fetal development. But by the time the baby is born, these nuclei should be around the edge. So when you do this stain and you see this pattern, we call that myotubular myopathy. And nobody in this room had this pattern, and that's why nobody here carries the diagnosis of myotubular myopathy. In contrast, over here, this is a stain that stains different fiber types. You may be aware that we have different kinds of muscle, and the easiest analogy to that is like chicken. When you have chicken, a chicken breast is the white meat. That's fast muscle fibers. They're used for large, quick, energetic bursts of energy when a bird flies. And the chicken drumstick is dark meat. That's slow muscle fibers. They're used for long, sustained contractions, for like standing around and walking, which the chicken does. Humans don't have specialized muscles like that, but we have a patchwork. And you can see that here, where these dark fibers are all interspersed in a checkerboard pattern of white fibers. These dark fibers are so-called slow, or the dark meat of the chicken, and the light fibers are the fast fibers. But in some patients, what you see is this, where all the slow fibers are much, much smaller, they're atrophied. And so we call this congenital fiber type disproportion. So it's a very descriptive name. It just simply means that certain fiber types, in this case, the slow type 1 fibers, are congenitally disproportionately small. So, and I'll just say that for some people with mutations of the tropomyosin 3 gene, or the myopathy genes, their muscle can, instead of looking like myopathy, can look just like this. So one of the points I want to make here is that there is a lot of variation in the way the muscle looks under the microscope. And although we use that microscopic result to give a name to the myopathy that somebody has, sometimes it pigeonholes people in ways that don't really correlate to what their genetic basis is. So the same genetic basis could either cause congenital fiber type disproportion or myopathy, and really essentially those two people would have the same condition. Where that's important is when we think about therapies, because if we have a treatment that's directed to the gene, then we don't really care what the muscle looks like under the microscope. Now that said, you're going to hear a little bit later today, I think, from Mike Waller, who Mark mentioned, who's a neuropathologist who trained in my lab. And Mike he's an expert at looking under the microscope, and so he'll tell you more about the importance of what the muscle looks like under the microscope. In the interest of time, I'm going to quickly skip through. This is something that I think many of you know. For the congenital myopathies as a group, there is as yet typically no good effective treatments that are directed at the underlying basis. There is, however, a lot that can be done. You're going to hear about that later today from Dr. John Bach, who's going to talk about pulmonary management. So that's extremely important in managing your breathing. And then you're also going to hear from Dr. Roy, who's going to talk about orthopedic intervention. And as everybody here knows, there's a tremendous amount that can be done to help support you and help you get around with that. So there's a lot that can be done in terms of managing the condition and really improving quality of life. But when I'm talking about treatment and what we're ultimately hoping to get at is by knowing the underlying genetic basis, can we come up with the specific approach that targets the underlying cause that will improve the muscle strength at a basic level. So this is kind of the genetic picture. This is a purposefully confusing slide, so don't worry too much about the details, but I'll tell you what the take-home messages are. This lists here a number of these congenital myopathies categorized according to the way the muscle looks under the microscope. This is the traditional way. So, Nevelin myopathy, central cord disease, myotubular myopathy, congenital fibrotectus purpura. These are descriptive terms. And what we found over the years is that each of these is what we call genetically heterogeneous. What we mean by that is that there is more than one genetic reason that could cause one of these conditions. So you already heard that there are 10 different genes, any one of which could cause Nevelin myopathy. And this is from 2011, this just shows the first 7 of them. There are a number of genes that can cause centronuclear myopathy, and so on. But if you look carefully at this list of genes here, you'll see something else, and that is that the same gene can cause different muscle pathologies. So the actin gene. We first discovered the actin gene as a cause of Nevelin myopathy. But it turns out that there are some patients when their muscle is down under the microscope that appear to be something called mini-cord disease. Others that have congenital fibrotectus purpura. And yet they all have a mutation of the actin gene. So again, not to beat a dead horse, but this is a really important concept to get, because when we talk about gene therapy, especially, or some of the other approaches that utilize molecular interactions or drugs that target specific proteins, we have to make sure that we're targeting a protein that's defective in a particular individual. And if any of you have been hearing anything about President Obama's Precision Medicine Initiative, that's what this is all about. Precision medicine is about being precise to understand the basis for somebody's medical condition, that individual person, and coming up with a treatment that's targeted specifically to the underlying basis. So just a few more examples. Tropomyosin 3 I mentioned earlier can cause any either of these. The RAND receptor mutations can cause a number of these different conditions. So this is you guys. This is you guys, and I'm pretty sure it's this exact same room. Was it this room earlier 7 years ago? And this illustrates, I love this picture, it illustrates a number of different things. One is that Nevelin myopathy in particular is probably the most common of the congenital myopathies. We think it accounts for about half. So we got a good turnout. We got a bunch of families here that can all get together. I already mentioned a little bit, it's very typical congenital myopathy, tends to be relatively non-progressive. Primary muscle weakness and hypertonia. The other thing that this illustrates is how variable this is. So some of these children, as you know, are probably not able to sit up on their own, certainly not able to get up and walk around. They require extensive ambulatory support. And then some of the other individuals in the back have been walking and having children and grandchildren and so on. So it's a very variable condition. Under the microscope, I didn't show you on the previous slide, but this is what myopathy looks like. And that is, this is a special stain called the trichrome. Normally, these fibers should just be uniformly stained, but you see these dark blue structures in this. These are the so-called monochrome rods. They don't look much like rods here, but if you look at the electron microscope, very, very high magnification. Here are the sarcomeres I showed you before. These are the Z lines, and what shortens is from. All right, didn't see that. What shortens is like this. Okay, that's the first time I've ever fallen off the stage. These things here now appear like rods, and these are the so-called rods. In fact, this is from an individual who was able to walk, and you can see there's a lot of organized muscle structure. So despite the fact that there are a lot of large abundant rods in the muscle, there's also a lot of organized muscle. So this person was able to walk. This is from a baby who was really quite weak and never was able to sit and walk. And even though the rods are smaller and more granular looking here, when you look at the electron microscope, you can see the muscle fibers are really very disturbed and developed. You had a question?
Yeah, I was wondering, this slide before, where you're talking about all of the, sorry, going before this, all the different things that it can be. So my son has the actor one. So would he have more in common with other people that have actor one, or more in common with the myopathy group?
Right, okay, so that's a great question. This is exactly what I was trying to get at. The answer probably depends on what you mean by more in common. So if you are asking the question of what can he do in terms of how much muscle strength he has and walking and running and needing breathing support or not, that is variable across all of these congenital myopathies. And you could have more in common with anybody with a mutation in any of these. There are generalities. So, for example, boys with myotubular myopathy tend to be much more severe. You don't have anybody with myotubular myopathy who grows up to have grandchildren. But clinically, there's a lot of variation. When you talk about a molecular level, from my perspective as a geneticist and somebody trying to think of a treatment, the answer is yes, you have more in common with somebody with congenital fiber defect disproportionate mutation in the actin gene than we would with somebody who has a nebula mutation. It depends, though, how we're thinking about the treatment. So Mike Lawler later today is going to talk about something called myostatin inhibition, which is an approach just to make the muscles bigger. And that concept should apply equally to anybody, regardless of their mutation, we think. In theory, is the idea. Because it's not targeting the underlying cause. On the other hand, if we come up with a gene therapy for actin mutations, well, that gene therapy would work for your son, and it might work for his friend with CFTD, but it wouldn't work for somebody else in this room who has a nebula mutation.
Okay. It's tricky.
Yeah, and that's why you need to think of it as different levels.
Right.
Okay, so here's our diagram that I showed you, and I showed you this. And this just I'm going to go through now what we know about a number of the genes that cause myopathy. So I mentioned actin, and actin is one of the first genes, actually the very first gene that's discovered was by a good friend of mine, Nigel Lang, in Perth, Western Australia. We have a couple of families here, I think, from Australia, and families have been diagnosed there. And Nigel was the first to discover that a mutation of tropomyosin 3 could cause myopathy. And there was a very special family in Western Australia where the tropomyosin 3 alteration had been passed down among multiple generations, and that allowed Nigel to find that gene and discover that tropomyosin 3 mutations could cause myopathy. So soon after that, Nigel and my groups together found mutations in the actin gene. And I should say that the tropomyosin 3 mutations are relatively rare. We think maybe 3 or 4% of individuals with myopathy have a TPM3 tropomyosin change. But whereas maybe a third of people in this room are going to have an alteration in the actin gene, like you saw. The next gene that was discovered was found by Karina Walker and Pedersen in Helsinki. And Karina's lab was studying nebulin. And nebulin is this very large protein, It tends to be mutated fairly often because it's so large, it's a big target. When random changes happen in their genome, they're more likely to affect the nebulin gene than if they are a tiny actin gene. And so therefore, we see more people with nebulin alterations that cause neuromyopathy. We think maybe up to half of people have neuromyopathy mutation in the nebulin gene. And then a few percent of mutations in a few of these other genes, one of the troponins. We've just found 3 individuals on this planet with mutations of this protein, lipoprotein called cofilin, and some others. So what this does, though, is it allows us to develop a unified hypothesis. Why is somebody weak if they have neuromyopathy? Well, I showed you what the thin filament does and how critical that filament is to muscle contraction. It stands to reason that if there's something messed up with that thin filament, you're going to have weak muscles. And since that thin filament is this complex structure with multiple proteins, it again stands to reason that mutation in any one of them could cause something very similar. It's like a carburetor in a car. You could have a clogged valve, you could have a broken spring, and you could have something else happen to the carburetor. All those different things result in the same thing: the engine stalls out. So I used to like to say to medical students, I'm not an MD myself, but I teach medical students, and I say to them, heterogeneity rules the day, and this is really a very common principle in biology. There's a lot of variation we see. Yes?
Well, back to the fibers, and why can't somebody just take a lot of protein in their diet, because protein builds muscles?
Yeah.
Or like in athletes, they take muscle building drugs, and they can get that won't work for the.
No, that's a great question, and in fact, there may be something to it. There are people that are thinking about how do you boost protein production in the muscle. But if you think about it, the basic instructions for building your muscle are defective when you have an alteration in one of these genes. And so you can build more of it, but you're building it from a defective plan. That's what the mutation in the gene is, essentially. And so with nebulin myopathy, we have partial function. So making more muscle that's partially functional can somewhat make you stronger, potentially. And there is evidence that some individuals with nebulin myopathy actually respond very favorably to exercise and workout training. That's not true for muscular dystrophy, but that's one area. It's not true for everybody, though. We don't completely understand it. It probably relates to the basis for the underlying basis that some people are susceptible to muscle building and others are not.
So basically, this theory would produce negative results and actually be a negative effect, right? Is that what you're saying?
I'm not saying that it would necessarily be harmful, but it might not help because you're building more defective muscle, in effect. But as you all know, there is significant residual muscle strength and activity. So, Mark can speak to this as a physical therapist. Building more of it may allow you to have increased function. With muscular dystrophy, there's a concern. There, the muscle dies and it regenerates and it dies and eventually it loses the ability to regenerate. So there's always been in the medical field a concern that for some patients with muscle diseases, having them exercise too much might speed up this progressive process. But in nebulin myopathy, as I said, the muscle is fairly stable, it sticks around, it just doesn't contract as well as it should. So we do believe that having more of it may be beneficial in some cases. And there are a few examples of this. There's one famous guy in Australia whose muscle nebulin myopathy affects only his fast muscles, and he has good slow muscle fibers. And he exercised so much he became a competitive bicycle rider. He actually could race competitively, but he didn't have any sprint capacity, so he was well ahead of the pack at the end. He did okay. That said, just anecdotally, it doesn't work for everybody, and so it's not something you can say, I'm going to exercise my way out of this. On the other hand, working with a trained physical therapist and your neurologist and your doctors, it's a question to ask. It's not something I'd recommend that you just go off and do on your own. Okay, one last point, and one basic point is that, as I said before, And again, this strikes on this theme that what the muscle looks like under the microscope isn't everything. And having them on rods isn't everything either. Here's the muscle from a dog, and it's hard to see in this picture, but there are some network rods in here. Turns out this dog doesn't have network myopathy at all. The dog actually is hypothyroid, has a canine hypothyroid myopathy. And so turns out that there are other insults to the muscle that can cause network rods to form. And these include some drug-induced myopathies, hypothyroidism. Some patients with AIDS, with HIV, they develop a muscle condition, weakness called HIV-associated myopathy. We see rods forming. So the point is that the Nibelung rods, even though they're used to help identify this condition, they very reliably occur when we have mutations in some of these genes. They're really a response to the primary defect in the muscle. So sometimes Nibelung rods can pop up in other situations. And in fact, if you looked at all of my muscles enough, or somebody here who doesn't have Nibelung myopathy, you'd probably find a few rods in a few places. So the rods are really a response to the weakness and to the underlying cause. However, when they occur in the presence of muscle weakness and one of these genetic changes we're talking about, that helps define what congenital myopathy is. So let me talk a little bit about genes and how we've identified a new group of genes besides the ones I was just showing you for the thin film. So my lab, many of you have been enrolled in my lab research study, and we started back in 1992. We now have almost 3,000 individuals and over 300 families with Nibelung myopathy enrolled, and this has been a tremendous help in allowing us to identify the genetic basis for this. And so I want to just take this second to thank everybody for their participation in research, not just in my lab, but in all of the groups. Everybody who's doing the natural history study downstairs, everybody working in Jim Dowling's lab, Karsten Bonneman's group, the CMDIR, and then our various other labs around the world. Anything you care to do can help move the field forward. And there's no need to choose one or another. You should be comfortable to participate in any and as many of the different research initiatives as you feel appropriate for yourself. And they all complement each other. We're a very nice community. We all cooperate because it's the only way to really make progress in a field like this. So genetics. So we already knew, for example, that mutations in the actin gene, the tropomyosin gene, and the Nebulin gene could cause Nibelung myopathy. So how did we find a mutation in the cofilin gene? Well, we knew cofilin were these purple dots. We knew from basic biologists who studied the structure of muscle that cofilin was important for helping to stabilize the actin-thin filament. So the function of these purple dots is to help glue that filament together. And we had a very interesting family in Bahrain, in the Middle East, where you can see there's a lot of consanguinity. There's a lot of first cousin marriages here, which is very common in that population. And as a result, there was a couple who had two affected girls with Nibelung myopathy. And the reason they were affected is that they had inherited the same abnormal gene from a common ancestor. And so by tracking down, we identified that in the region that they shared in common, that they had both got from their ancestor, was this gene for cofilin. And knowing ahead of time that cofilin was important for holding the muscle fiber together, that allowed us to go in and do DNA sequencing and sequence the cofilin gene. And I won't go into details, but this is the sequence tracings that shows that, yes, indeed, these two girls had an alteration in their cofilin gene that affected the protein shown schematically here, an altered amino acid at position 35, and that in fact was the cause. So when we have families like this, and we know something ahead of time about the structure of this, we can go in and ask the question, are there mutations in genes that encode some other components of this? So again, getting back to the carburetor, it's like after saying, okay, there's a new spring here I just discovered. I'm going to go look now and see if that's broken or not. Then this is the actin gene. The point to take away from this is that it's 3,561 bases, 3,500 bases. So to give you a sense of scale, our entire genome is 3 billion bases. So the actin gene is a very, very tiny part of our whole genome. Because it's tiny like that, we can actually go in and sequence it fairly easily. And that's one of the reasons why we found it's been fairly straightforward to identify the group of people who have alterations in our actin gene. We don't have that much to look at. In contrast, this is kind of fuzzy and it's incomplete and can't really see much, but this is a schematic of the nebula gene. And the point is it's big and messy. And whereas actin is 3,500, this is 250,000. So orders of magnitude larger. That's one of the reasons why, although we suspect that maybe half of all individuals with myelopathy have an alteration in their nebula gene, it can be very difficult to actually find those changes because we can't go in and sequence directly all 250,000 bases the way we do the 3,500 in actin. So there's a new technique that some of you might have heard of called next generation sequencing. This is also a method that it's used to do a couple of other things you may have heard of. WES or WGS, that stands for whole exome sequencing or whole genome sequencing. So if you recall the Human Genome Project, back in the 90s, it took 10 years and $3 billion to sequence the first complete human genome, to create a sequence of all 3 billion of our bases. And in fact, even when they said it was complete, it wasn't all 3 billion. It was a good percentage of them, it was most of them, but not all 3 billion. So that's a genome. Then, if you want to ask, well, genetic conditions like myelopathy are typically caused by alterations in protein coding regions. Turns out that's a much smaller part of our genome. We call that the exome. It's the part that's expressed and turned into proteins. And so that's what whole exome sequencing does, looks just at that. So I'm not going to go through the technology here, but suffice it to say, it utilizes advances in photolithography, which is used to create computer chips, to make arrays with large numbers, hundreds of thousands of different molecules on them. And then the machine shown here actually could fit on this podium. So it's really incredible today, because now in 2 weeks or a month, with a machine that you could almost lift with 2 people, you can sequence somebody's, all their protein coding regions, their exome, or even their whole complete genome. And that's revolutionized the way we look for these changes. This illustrates how we map that. I'm going to skip over this in the interest of time. But I want to tell you about some of the things we found using this new technology. So Nanda Gupta, who trained in my lab and who unfortunately cannot be here tomorrow, although her name's on the schedule. Vandana was a postdoc in my lab, now has her own independent lab, is working across the street at Brigham and Women's Hospital. She's an assistant professor. It's always fun for me to see people going out on their own. But what she did, in collaboration with some of the people I've mentioned, Nigel Lang, I think Karina Walker-Peterson is on here somewhere. There she is. Vandana took the DNA from a group of people with myelopathy who we hadn't known what their genetic cause was, and used whole exome sequencing to sequence all their various genes. And she discovered a group of them, 5 children, who had congenital myelopathy, and where their muscle had no neurons in it. And she had mutations in a gene called KLHL41. Again, we have to give everything a name. The names don't always make sense, and so it gets to be these confusing acronyms. KLHL41. These 5 kids have pretty variable degrees of weakness. So fetal akinesia, akinesis is movement. Akinesis is a lack of movement. So akinesia, fetal akinesia is a fetus that didn't move. And it didn't move because it was too big in the uterus. And if someone is born, it's often got contractures. And so the most severe of these children had that condition, and they died very soon after birth because they were so defined weak. And yet some of the others had delayed ambulation, but they walked. And the oldest one was 12 years old and still walking. So again, a lot of clinical variability. And what Vandana found was that the severity correlated with the mutations effects on the proteins. So the most severe children made no protein. They were completely missing this KLHL41 protein. And the ones who were able to walk had a different mutation that alters the protein, reduced its function, but didn't completely destroy it. And so that's another feature. And so, for example, among the patients with actin myopathies, with variation in the actin gene, some kids are very weak, others are relatively not so weak, and many of them can grow up and have grandchildren, like I've said. And so it's correlated with how damaging that particular mutation is on the protein. This is actually one of our dilemmas, because many of you, when you first get a genetic diagnosis, you probably ask, so what does this mean for my child? And the doctor probably had to say, well, we're not really sure. And unfortunately, that is still the case in a lot of these instances, because many of the changes we see is the first time we're seeing them. But as we gain more experience, as we increase the numbers, and this is where, for example, natural history studies are important, we can start to track and we start to see recurring patterns. And then hopefully, in the not too distant future, somebody gets a new diagnosis and we'll be out there and say, okay, your change means the following. This is what we can expect. This is what we can do to try to prevent some of the negative things from happening. And this is how we can hopefully treat it. Again, that's what precision medicine is all about. So this just shows that the mutations that she found is spread throughout the gene. This is what the muscle looked under the microscope. You can see very typical rods. So just looking at these patients, there's nothing that a neurologist could tell you they were any different from anybody else in the room. There was nothing that Mike Lawler, the neuropathologist, could tell you would be any different from anybody else. They were really typical neuromyopathy, just like anybody here. But the mutation was in this thing called KLHL41. And I haven't told you what it is yet, but for the moment, I can tell you it's not a piece of the thin filament. So this just shows that the protein is actually missing. And in some of these patients, or greatly altered, it's very hard to see very thin, but these are black, and that's because there's no staining of the protein here. So Vanity used zebrafish to try to prove that these work. She'd only found it in 5 people. It was the first 5 people in the world. So it could be that maybe some people on this planet just simply don't make KLHL41 and it has nothing to do with neuromyopathy. So to try to prove that these KLHL41 mutations really cause neuromyopathy, she used zebrafish. The first thing she did, turns out zebrafish are kind of unique, that oftentimes they have 2 genes for every one of ours. There's been some genetic duplication. But the 2 genes both largely do the same thing in a fish muscle that same gene does in our human muscles. So it was the A gene and the B gene, and she stayed developing zebrafish. You see this chevron striped pattern here, that's the developing muscle of these fish. So this told her that these proteins, KLHL41, are used mostly in developing muscle. So that's encouraging. That's consistent with our thought that neuromyopathy genes would have to be in muscle. And then she did something very powerful that zebrafish allows us to do that we could never do in human, and that is actually create neuromyopathy by altering that gene. So what she did is she turned off the KLHL41A gene or the KLHL41B gene, or both together. Because the normal fish, it's nice and straight at 2 days in life. And here is this fish where they've both been turned off and it's kind of curled up a little bit. And one of the nice things about zebrafish is while this fish is still alive, you can look at it under the microscope and look at the structure of the muscle using special optics. And you can see a very bright pattern here. This is normal muscle because it's highly organized, and so it's glowing under this special optics called birefringence. And when neither of the A or the B genes will turn down on both, you basically see that glowing go away. And what that tells you is that even though there may be some muscle here, it's very highly disorganized. So turning down or turning off this new gene created disorganized muscle very similar to what we expected in neuromyopathy. I'm afraid you can't really see this very well in the lighting, but this is actually looking at the muscle under the microscope at higher magnification. And normal muscle has a very highly organized pattern, and you can barely make out some stripes there. And then if you're not going to use the A gene, the B gene, or both of them, the pattern of the muscle fibers is highly disorganized. So if you think about, like, if you have salmon, you know, flakes of the fish, that's what each of these little chevrons is. You can see it. And in these fish with the KLHL41 knockdown, those flakes will all disorganize, and that affects the effect of the muscle. This is looking at it in the electron microscope, so higher power. And again, this is the wild type, the normal muscle, and you can see when you knock down A or B or both, you get all kinds of disorganization. And in fact, these are the Z lines here. You see this dark structure. Looks like the beginning of what we would consider to be an emerald rod. So pretty good evidence then that this gene is involved in emerald male. So I told you that this protein KLHL41 is not part of the thin filament. So what is it? And why does its alteration cause an emerald? Well, here's our structure of the muscle again. The Z line is here. And then this is the thick filament, the myosin, the thin filament. And what we found when we stained with antibodies to KLHL41 is not present in here. Instead, it's present at the periphery of the myofibrils, the contractile apparatus, in a structure called the sarcoplasmic reticulum. And this is an area where proteins are made and processed and stabilized. So it makes us think that maybe this has something to do with making proteins. So what is KLHL41? Well, to step back for a second, to put this in context, it turns out that there are 3 of these genes with the letter K in their name that we found can cause myelopathy. KBTBD13, KLHL40, and KLHL41. The K in each of these refers to the word Kelch. And so it turns out in our genome there are 63 different genes that have Kelch as part of their structure. Kelch it's a part of the protein, the so-called domain, that is responsible for binding other proteins. So it doesn't tell us what it does, but it does just tell us that this is a protein, interacts with and binds other proteins. And one of the things is that many of these genes, most of these genes, their function is not known, including KLHL40 and 41. But some of them, the function is known, and it's known that they bind to something called column E3 ubiquitin ligase. So again, a lot of complicated, messy name, but we break it down there. So ubiquitin is a small molecule that gets attached to proteins as they get older. So our genes are always being turned on and making new proteins. And as time goes on, the proteins age, they get degraded and damaged, and then they get broken down and recirculated. And our bodies are constantly renewing like this. And so the cell has a very highly organized mechanism for recognizing old damaged proteins that just occur over time. You go out in the sun, or you have oxidative damage. And when that protein gets damaged, then it gets marked for degradation and it gets replaced by new synthesis, the vascular synthesis of proteins. That's one of the reasons why to do that. So the way that the cell knows a protein is old is this column E3 ligase is a protein that attaches ubiquitin to it as it gets old and damaged. And then when it's old and damaged, it gets chopped up and recycled. The protein's components are recycled. So our model then is that KLHL40 or KLHL41 or KBTBD13 is a protein shown here that attaches actin or nebulin, one of the proteins involved in nebulin myopathy, to this called 3 ligase. So you can have nebulin myopathy because you have an abnormal nebulin or abnormal actin in the cell, messes up the cell. Or you can have mutation adapter protein. You have normal actin or normal nebulin, but as it ages, it gets damaged. becomes less functional. And yet, the mechanism to mark it as old and damaged is defective. It doesn't get derivative and added to it, so it doesn't get degraded. And so you get an accumulation of defective nebulae. And that's just the same as having a mutation in the gene in the first place. So I'm going to show one more slide and then a summary, and then we can honestly do some time for questions. This just summarizes our genetic studies in this group of families that involved in our research. And one of the things we're doing is using this next generation of sequencing to try to comprehensively sequence everybody possible and get a good handle on the relative contributions of different genes. So we find that nebular mutations are present in about 40%, 30 to 40% of patients we studied. And the actual number is probably closer to 50, but because we haven't been able to sequence the whole gene so big, we'll probably be sequencing those. Active mutations about 30%, 30 to maybe a third of families. Troublomyosin 3, about 3%, troublomyosin 2, and then all these other proteins about 1 or 2%. And then again, just to summarize, all of these proteins, nebulin, actin, TBM3 to troponin T1, cofilin, and something I haven't mentioned yet, like L-mod, myoglobin 3, all of these are parts of that thin filament. So the unifying theme here is that defective thin filament. And then we have these other 3 genes, KLHL 40, 41, UKBTBD13, and these are probably involved in the machinery specifically to turn over these thin filament proteins. So, as a scientist, it's satisfying, but it also has real practical implications because it means when we start trying to think about therapy and treatments, we know what to focus on. This is the area that we have to think about. And anything we can come up with to alter these patterns. And tomorrow, I'll tell you a little bit about how Babylon is using drugs that are known to enhance protein turnover as a way to possibly treat patients with nebular mutations. So having this information sounds a little bit dry and esoteric in some ways, and kind of really crappy names for the genes, KLHL. But if we really drill down to what it all means, it can actually have big implications for how we come up with treatment. Everything around. Okay, so let me just go back and end with the, thanks Mark, I'm not going to need it again. We'll end with our animation here. And the question was asked earlier about mechanisms of weakness. And just to remind you, normal muscle, this is what you saw before, we have nice contraction, we have thin filaments all of equal lengths. Tomorrow, hopefully, Kuhn Ottenheim will be here. He's flying in today, and he has done a lot of studies in collaboration with my lab, Siegfried Labate, who I think may be here, Mike Lawler, and so on, to actually understand the mechanisms for why the weakness is occurring. And I'm just going to show you for a few of these. So, as Mark mentioned, patients with nebulin mutations, the nebulin is one of the things it does is responsible for controlling the length of the thin filament. And when nebulin is mutated, some of the filaments are too thin, and you can see there's a lack of overlap. So it's not the rods, not these nebulin rods that are actually causing the weakness, but it's the fact that the myosin can't grab the thin filament because it's too short. And they're variable, so it's more disorganized. In contrast, if you have a mutation in tropomyosin, it doesn't affect the length of the thin filament at all, but it does, tropomyosin, remember, is responsible for blocking the actin and the myosin interactions. And so when you have that mutant tropomyosin, everything, the structure is there, but the grabbing and pulling is less efficient. And finally, actin can be one of the most devastating. And this is from somebody with an actin mutation who has a particularly severe form of the disease. And in fact, what happens is that the thin filaments actually just break down and don't retain their structure, and so then you get weakness. So I will stop there. I went a little longer than I hoped to, but would love to spend as much time as you guys want answering questions.
First of all, thank you very much for the presentation.
I'll pass this around if you like.
Sure.
Okay. First of all, thank you very much for the presentation. It seems like we've come so far with the genetic studies. And I'm just wondering, you know, most of us in this room, we've been diagnosed through a muscle biopsy. And I'm wondering, now that we've gotten so far with genetics, do you think that will ever come a day where we'll have like a panel? I know a lot of the parents are starting to talk about this. Can we get a panel when they see these young kids who are floppy baby or hypotonia, and they can look and test all these various genes for all the different muscle diseases through a blood test. And then they'll know, oh, look, there's a mistake in the nebulin gene. You must have nebulin myopathy, as opposed to, and how close do you think we're?
Yeah, so we're in a transition, we're in an interesting transition phase, and actually, you know, one of the other hats I wear is I'm in the genomics division, and I run a project that is doing whole exome sequencing on healthy newborn babies, actually both healthy and sick newborn babies. So we're testing this out. If you sequence all the 20,000 genes in a baby, what can it tell you? And in particular, if that baby's in the NICU and is floppy and has muscle weakness, if we find a mutation, that can often allow us to tell. And in fact, this discussion goes back years, for example, when we first discovered the mutations that cause Duchenne dystrophy. Because if you have a question about that, it's very easy to find a Duchenne dystrophy mutation. It's a deletion. If you find that at the DNA level, you probably don't need the muscle biopsy. I would say that there are going to be some genetic variants that are very difficult to understand, and it's always going to require muscle biopsy. I think in a lot of cases, the risk of muscle biopsy is done because this type of panel or whole exome sequencing isn't available. But also increasingly, it's going to start to be done. Now, clinically, one of the things I tried to get across is that the congenital myopathies, there are a large number of different genes, and other genes besides the ones that cause congenital myopathy that in the clinic, the patient looks the same. So when you have a floppy baby or a weak baby, there's lots of causes, including some that are not congenital myopathies. So it can be difficult to know how to test. In the past, we had to assume we'd have to test for one gene at a time, and then the muscle biopsy was critical. Now we're having tests that look at like 40 different genes at a time, or 60 genes at a time for all of these panels. Or there's the whole exome. And depending on your index of suspicion for how to categorize that. So it's a pretty wishy-washy, unsatisfying answer, but it's happening.
Get there. Okay.
I wonder, do you have a sense of how many, what percentage of mutations you're capturing with the whole exome sequencing?
Yeah, so traditionally in genetics, the rule of thumb is that if you have somebody with an unknown genetic condition, anything, not just congenital myopathy, and you do whole exome or whole genome sequencing, we've been making diagnoses about 1/3 of the time. So that's pretty disappointing. I mean, it's wonderful for that 1/3, and actually, that's actually tremendous advancement from where we were 5 years ago. For nebulin myopathy, we've found mutations in approximately 80% of people that we have exhaustively studied. The remaining 20% we can't. Now, I believe that nebulin myopathy, I believe they have an alteration, but it may be of a type that's not findable by the current technology. So one point is that the sequencing that we're talking about looks at the DNA at the very finest of levels, at the level of A, C, G, T, each of the 3 billion bases. But sometimes the mutation can be so large that you don't see the forest for the trees effect. That if it's, because you've got the gene here, you have a whole section of it that's been taken out and inverted, that'll disrupt the gene. But when you sequence the tiny little pieces in isolation, you can't tell. And that's the type of mutation that we're currently missing. We're thinking it's 10-20% at this point.
And so do you have strategies to try and go after those mutations?
Yeah, so nowadays, the type of sequencing I've been talking about is being supplemented with either chromosome microarray or insertion deletion testing, looking for, the other thing is that we may not, everybody has 2 copies of everything. Suppose you have a deletion that removes one complete copy. You can't really tell when you're sequencing the DNA, but there are ways to find that. And so those are picking up another 5%. We still have 10 or 15% though that we can't figure out. Some of those mutations are going to be outside the gene. Some of those are probably genes that we may see them in our computer files, because we got a computer file with 200,000 entries. And the question is, which one of those entries is the mutation that caused the disease? It may be in a gene that we don't recognize, that we haven't learned enough about to know that it can cause an abnormality. And I'm sure there are other genes, like these Kelch genes, that we just haven't found yet that affect the film development. So that's the more slow process, but something we're very actively working on.
Just real quick, I wanted to add, my son Gavin is 8, and when we were first tested for nephropathy of the nebula gene, it was negative. And within a year, they said they got approval for further testing, and he was actually positive for the nebula gene, so they were able to find it. So I think the advances that they're making are just excellent, because now we have the specific.
Yeah, and it's continually improving. I mean, for example, in particular, the nebula gene, as it's very long and complex, it has these repeating units that give it its size. There's one region where there's 3 segments in a row that are almost identical to each other. So when we sequence them, we're actually sequencing all 3 of them simultaneously. We can't tell them apart. So if there's a mutation in one of those 3, we can't see it by the traditional method. But now we are starting to come up with ways to overcome that. So as we learn more about it, our sensitivity is increasing. So never take no for an answer.
This may be a little more involved, but so the actin and myosin complex of the sarcomere in how the muscle contracts. In the cardiac muscle, there's actin and myosin in the sarcomere and cardiac contraction. Why is it that nemolymyopathy doesn't affect the actin that is put into the cardiac muscle, but it does in this skeletal muscle?
Great question. And actually, understanding that leads us to therapy. So it turns out that we're so much specialized. And there's one gene that's turned on to create skeletal muscle actin. It's called ACTK1. And then there is a different gene used in heart, and it's ACTC, cardiac one, ACTC. So, in fact, in very early development in the fetus, in the skeletal muscle, both these genes are turned on in the skeletal muscle, and then the cardiac gene gets turned on. And so some rare individuals who have completely devastating knockout mutations of the skeletal actin gene, those babies are born because their muscle contains cardiac actin. But it gets normally turned off in the skeletal muscle, remains on in the heart. There is a little bit, but very little of the skeletal actin gene turned on in the heart. And that's why you can have a mutation that affects your skeletal muscle, but not your heart. Unfortunately, almost everybody, I'll never say never, and never say nobody, but as far as we know, mutations in the skeletal actin gene don't cause cardiac disease. In contrast, this cardiac gene, ACTC1, is a well-known cause of dilated cardiomyopathy. So, cardiomyopathy is kind of this heart equivalent of neuromyopathy in the skeletal muscle. It's because of different genes, that's the short answer. Some of the proteins I've talked about are used in both heart and muscle, the skeletal muscle, and mutations of those actually lead to a neuromuscular condition that includes also cardiac problems. Mark, you want to
I was going to say that we've run a few studies, I don't know, maybe later time, but United Planning in Australia. He's actually trying to look at how you modify the gene profile of the cardiac muscle within nemolymyopathy and make it help to improve cell.
Right, because I was just curious because it's electrically stimulated cardiac muscle. Is there a study that shows whether or not electrical stimulation on the muscle fibers of nemolymyopathy patients are actively given therapy for cardiac muscle?
Yeah, I think it would improve the electrical firing, but and support electrical firing, that doesn't necessarily mean that the proteins will reach in simple format. My last question is, I insert this one purposefully just so people in the room can hear. How much of your time do you spend actual research, bench things, trying to find more ingredients, trying to come up with treatment? How much time do you spend fighting for dollars so that you can do these things?
Well, so I have ADHD, so I'm sitting at my desk, I'm kind of doing 3 or 4 different things at the same time. And some of it is actually looking at data and analyzing. But unfortunately, in today's financial climate and the way that government funding is going, and the Muscular Dystrophy Association has supported me for many years, but they've let me down on hard times. I'd say I spend probably 2/3 of my time thinking about how to get funding and managing the funding I have. Write progress reports, which sometimes are onerous to me, but they are actually very important. And importantly, a lot of what I do is disseminating information. And it's like a tree falls in the woods, and nobody hears it, it will fall, or whatever. It doesn't make any sense for us to do work in the lab if we don't tell people about it, we give people the benefit of it. So a lot of my time is spent on things. But it's amazing. So, Patty, do you want me to chat with you?
Actually, I was just going to say thank you very much, Dr. Baggs. We appreciate your talk. So let's give him a round of applause. At this time, we're going to take a short break. We'll come back and we'll do some family introductions at 10:45. We're going to end the live stream right now, but we will restart it again at 11:15 for Dr. Baggs' talk on respiratory. Thanks.
And I will be around, so anybody, please grab me anytime you like.
Thank you so much.
I was trying not to go too long there.
Oh, you
I got to smoke it for another hour and a half.